It's my pleasure today to introduce Dr. Nicola Palomero-Gallagher. Dr. Palomero-Gallagher graduated in 1990 from the Sciences faculty of Universidad Autónoma de Madrid, Spain. She was a PhD student at the CNO Vogt-Brain Research Institute of the Heinrich-Heine University in Düsseldorf, Germany, between 1992 and 1999. And she received a PhD from the same institution in 1999. She is a senior researcher and the leader of the Receptor research group at the Institute of Neuroscience and Medicine in the research Centre Jülich in Germany. And she's an associate professor at the CNO Vogt-Brain Research Institute of Düsseldorf University. Her research aims to shed light on the relationship between structural, so cyto and fiber architecture, molecular, so receptor architecture, and functional organization principles of the brain. Please join me in welcoming Dr. Palomero-Gallagher.
Yeah, no, it's okay. It's dangerous to put a liquid near my laptop. I've managed to destroy a couple of keyboards, and I don't want to do this during the talk. Okay, so thank you very much for the very kind invitation, and also for so many people being here in presence, although I know that it's a midterm break, and also for those of you who are on Zoom. I'm going to tell you something about my research stuff that I've been doing like over the last 20 years, at the beginning with a bit of an introduction to the principles of what we're doing, and then some new stuff that one of my postdocs is doing. So I'm particularly interested in receptors for classical neurotransmitters because they are the link between the brain's structural segregation and its functional segregation, okay? And when people think about connectivity in the brain, they normally think about fiber tracts and axons, right? And we know that within a neuron, along these axons, signal transfer happens via electrical conduction, so what we call the action potentials. But once this signal reaches the end of the axon or the end of the fiber tracts that are in the brain, there's a space. There is actually no physical connection between two neurons that are communicating with each other, okay? And when you reach this point here, the synapse, then you have structures that are zoomed into here on the right of the slide. This part here is the so-called presynaptic part of the synaptic contact, and it belongs to this neuron here, which is sending the information, and this bottom part here belongs to the postsynaptic component, and it is the receiving neuron, okay? And these two parts, there is really physical space between them. It's very, very small, but there is a so-called extracellular space where we've got the synaptic cleft. So in order for the information to be transferred from the sending neuron to the receiving neuron, the sending neuron has to release chemical substances, which are called neurotransmitters and are highlighted here with these little red balls, okay? And these chemical substances have to bind to another structure, which is called a receptor, and which is embedded in the membrane of the receiving neuron, okay? And I've got different colors here because there are different kinds of receptors, and I'll go into a bit more detail about this later on in my talk. Now, these receptors are simply proteins or protein complexes, okay? And we've got different methods that can be used to visualize them. I've got a couple of examples on this slide here. On the left, there's positron emission tomography, which has the enormous advantage of being able to be used in vivo. You can see time-related changes it's got the disadvantage that it's got a relatively low spatial resolution, and also due to ethical constraints, there's a limit to the amount of compounds that you would be able to visualize in one single individual. And then the two methods on the right of the slide are both post-mortem methods. So that's one of their disadvantages, that you have then a lot of technical issues, and they're timely, demanding, but they provide a higher resolution. Now, what's the difference, basic difference between the immunohistochemistry and the receptor autoradiography? The main difference is that with immunohistochemistry, you've got antibodies that specifically bind to a single type of protein, okay? And you don't know if, in the case of the receptors, when they're protein complexes, you don't know if that protein that your antibody has bound to, if it is really part of a membrane-bound active receptor, as in the example here in this slide, or if it is if it's a protein that has just been synthesized, so it's actually still somewhere in the cell body, it hasn't been transported out to the synapse. So at the moment of death, it wouldn't have been able to influence the activity of the neuron that's expressing it, okay? That's the big disadvantage of the immunohistochemistry. The big advantage is that you really have an impressive resolution, especially if you use electron microscopy immunohistochemistry, and that's what this image here shows, where then the presynaptic neuron, the sending neuron, is highlighted in yellow, and these dots that you can see here, the red and the blue coded dots, they are subunits of different receptor types, okay? Receptor autoradiography, in contrast, has the enormous advantage that what you are actually labeling is the protein complex that at the time of death was embedded in the cellular membrane, and it would have been able to bind to a neurotransmitter, and it would have been able then to induce basically changes in the membrane potential of the receiving neuron. Okay, so this is the big advantage of this method, right? Now, in the example that I showed you before, I had one receiving neuron, and I had the axon of one sending neuron plus the neurotransmitter system. But in reality, the brain has very many different kinds of neurotransmitters, okay? And I'm just listing a couple of examples here: glutamate, GABA, acetylcholine. These are all so-called classical neurotransmitters. And each one of these neurotransmitters can bind to different types of receptors. These are these different colors in these symbols here, okay? And this list, it isn't a fully comprehensive one, it's just a couple of examples. And the reason why it's important that we've got different kinds of neurotransmitters and that each one of these neurotransmitters can bind to different kinds of receptors is because these receptors can be distinguished based on their mechanisms of action. So if activation of the receptor results in an enhancement of the activity levels of the receiving neuron, or in a dampening of its activity level, so if it's an excitatory or an inhibitory receptor, and also the mechanism of action. If you've got ionotropic receptors, which are actually ion channels, and they induce very fast changes in the receiving neuron, or if you've got a metabotropic receptor, which starts a cascade of events which ultimately ends with the opening of ion channels, but because this happens over a second messenger mechanism, it's a slower mechanism of action, it's more long-lasting. So this way, with these different types of neurotransmitters, different types of receptors, this gives the brain an impressive degree of plasticity by which it can modulate the way information is transferred, because it can send on the message, you know, change your activity level immediately, or change it slowly, but maintain that slow change over a certain amount of time, etc. Okay, so these are all different methods. Now, in the brain, neurons aren't isolated. They form parts of networks, okay? So, of course, a neuron isn't going to receive input from one other neuron. It's going to receive input from very, very many other different neurons, okay? And these different neurons don't necessarily all produce the same kind of neurotransmitter. They can produce different kinds of neurotransmitters. So that means that every neuron in the brain must be able to express different receptor types on its membrane, okay? The types of receptors which are expressed on a membrane, or the number of different types of receptors that are expressed by a neuron, can vary depending on the brain region you're in or on the cell type that you're looking at, but every neuron can express many different types of receptors. And this is what basically enables, you know, integration of information in the brain, right? Receptor autoradiography, which is the method that I'm going to be talking about most today, requires unfixed frozen tissue for for their visualization. And what you see here is a video of how a section is created in a cryomicrotome. Down here you have the frozen slab of brain tissue, and this is transported slowly but with a constant rate by the cryomicrotome. Here you have a knife which is super, super sharp. It can even cut brain. It can cut brain tissue. Yes, it can cut bone, which is what I want to say. It can cut bone tissue, okay? And then you have I'm going to start the video again. You here have like a plastic cover, which basically the section is sliding between the knife and this plastic cover, and then the technician has to very, very carefully slide the section onto the glass slide. It's a cryomicrotome, so all of this happens at below freezing temperatures. Last weekend in Montreal, you could have done it outdoors, but normally it's in this freezer device. We section the brains at approximately minus 20 degrees Celsius, okay? And then the sections are thaw mounted onto the glass slide so that they can be processed for visualization of cell bodies or of myelin or of different receptor types, okay? So once you have thaw mounted the section, that's it, it's going slowly. This little black box here is actually the video that you just saw now, but for today's presentation, I made it larger. Now we serially section the brains, okay? So that from any given rostrocaudal level in the brain, you can obtain very many different sections. Each one of these sections is 20 micrometers thick. Okay, you can go down more. I've also done 10 micrometer section or thick sections. But this means that for monkey brains, for human brains, you can acquire sections that almost, you know, they are neighboring sections, but basically so little has changed in the anatomy that you know you're analyzing the same structures. We then incubate these sections with either a radioactively labeled ligand. That would be this schematic representation here on the left, where we only have the red stars, or in incubation buffer, which has this radioactively labeled ligand and a so-called displacer, which binds to the receptor actually with a higher degree of affinity than the radio-labeled ligand itself, because often adding a radio-labeled isotope to the chemical substance that binds to the receptor changes the shape of that chemical substance, even if it's just a very small amount, but that already changes the affinity with which the ligand will bind to the receptor, okay? We also have a rinsing step to stop the binding procedure. We then expose these radio-labeled sections against films, which are sensitive to radioactive emissions, okay? And what you see here is you've got these squares. They're scales which have known concentrations of radioactivity and which have been calibrated for brain protein tissue content. And then on the top of the image, you've got autoradiographs which have ensued from this total binding experiment. And if you look very carefully at the bottom of the slide, you will see like ghost images. These ghost images are the result of incubation of the sections in the non-specific binding incubation, okay? So everything that you can see here in these ghost images, those are receptors in the brain tissue that have been labeled by the ligand, but they're not the receptors that you want to look at. It's the non-specific binding, okay? And we can then just subtract these values from these values, and that's how you get the specific binding, okay? We have to digitize these images for their densitometric analysis. So we've got an imaging processing system, we've got a digital camera, and we've got a source of homogeneous light. And that's how we get these images here, the standards. We've got a digitized receptor autoradiograph, and we can measure the gray values in these standards, and we can create a regression curve, which expresses the relationship for every gray value in these 8-bit images. So we've got a total of 256 gray values, the relationship between the gray value and the receptor density in femtomole per milligram protein, okay? And this is, as you see, it's a nonlinear relationship. And we can then transform every single gray value from every pixel in an autoradiograph into a receptor density in femtomole per milligram protein. And that's how we can measure our receptor densities. And because to the human eye, it's difficult to see differences in gray values when you've got this continuous range of 256 gray values. We divide them into a group of 11 discrete colors. So it's just like a linear enhancement. And that's how we get these color-coded images. And that's the kind of images that I'll be showing you then in other slides during the talk. So here what you see are coronal sections through a human hemisphere at the level of the central sulcus. So that here would be the central sulcus, and here you'd have the lateral fissure, and warm colors code for high receptor densities, and cold colors code for lower receptor densities, okay? And I'm going to, so already at this low resolution, you can see that receptors for classical neurotransmitters, they're heterogeneously distributed throughout the brain. You've got some parts with very high densities, other parts with low densities, depending on the receptor type that you are analyzing. Maybe the receptors highlighting basal ganglia or subcortical structures. So for example, here, the dopamine 1 receptor, their densities are very low on the cortex, but we've got these very high densities in the putamen and caudate nucleus. If we look at the serotonin receptor, for example, this is a neighboring section. So there actually is a caudate nucleus and a putamen here. It just has such a low density of 5-HT1 receptors that you can't see them, okay? Now I'm going to zoom into some of these sections to point out some principles that we can use for our research. And the first principle is that these differences in the densities of receptors, they reveal borders between cortical areas. I've serially sectioned the brain, so some of these sections have also been processed for the visualization of cell bodies, right? So I know that this structure here is the Heschel gyrus. It contains the primary auditory cortex. And I can see here that, for example, for the muscarinic M2 receptor for acetylcholine, it's has a very, very high density in the primary auditory cortex, and this density is actually significantly higher than that of surrounding secondary auditory areas, okay? And the point at which the densities change significantly is highlighted by these arrows. Now, if I look at a neighboring section, but one which has been processed, for example, for the visualization of the noradrenaline alpha-1 receptors, you will again see the Heschel gyrus, and here we've got the opposite situation. It has a lower density of alpha-1 receptors than the surrounding areas. But if you compare the position of the arrows, which highlight the place at which these density changes occur, in a significant manner, you will see that these changes happen at comparable positions. Okay, so when two or more receptor types show a border of a given area, this happens at a comparable macroanatomic or even mesoanatomic location, okay? But importantly, for brain functioning and for network building and so on, not all receptors show all possible borders in the brain, okay? And here I'm showing you an example of the NMDA receptor, which for the case of the primary auditory cortex doesn't really reveal the border between primary and secondary auditory cortex, okay? And in other parts of the brain, it can be other receptor types that don't highlight borders, okay? So yeah, not all receptors show all possible borders, right? And this is very important, as I said, for networking in the brain. Now, another organizational principle is one that you would see when you zoom into the area of the central sulcus, because here we know that the central sulcus is the home of the primary somatosensory cortex, which is located on the posterior wall of the central sulcus, and it is this part of the section which I have labeled with S1, somatosensory, and the anterior wall of the central sulcus has the primary motor cortex. So that's what we've got labeled here with M1. And if you look at the muscarinic M2 receptors and the alpha-1 receptors, and now just the serotonin 5-HT2 receptor as an example, you will see that that we have really big differences in the receptor architecture of the primary somatosensory and the primary motor cortex. So areas from different functional systems differ in their receptor architecture, okay? That's the second organizational principle. The third one that I want to tell you about is the fact that within a given system, differences in receptor densities can highlight different hierarchical processing levels. And now this slide doesn't just show the coronal section, through the region of the central sulcus, but also a coronal section through the occipital pole, where we've got the primary visual cortex. And if you look at these two sections, you will see that the primary visual cortex, the primary auditory cortex, and the primary somatosensory cortex are all characterized by very high densities of the muscarinic M2 receptor. And when you move then from the primary area, so the first part of the brain where you actually become conscious of the fact that you have seen or felt or heard something, right? When you move then through processing levels of higher hierarchical levels where more and more information becomes integrated as to, for example, if you're in the visual system, it's these parts of the brain are telling you, yes, you saw something, and you saw something that is static, and you saw something that is in color, and you saw something that is a face, and so on. So you're moving up through different processing levels, okay? So that's another important aspect of receptor distribution patterns that we can use to analyze the relationship between structural and functional segregation. And then another important fact is that these principles, which we have demonstrated like over the last, say, 20 years or so in the human brain, that they are actually, a lot of them are evolutionary conserved. So we can also find them in the brains of different mammalian species, and I'm here showing the macaque monkey and the rat as two, examples of commonly used animal models in neuroscience. And here you see the primary visual cortex in the human brain, the macaque monkey brain. You can see that in both species, you've got a very high overall density in the primary visual cortex. When we move to the rat brain, we also see that the primary visual cortex has higher densities of the muscarinic M2 receptor than adjacent cortex. But here, these differences, they don't happen across the entire cortical depth, but they're restricted to layer 4, okay? So depending on what neurotransmitter system you're interested in, you should be careful as to what animal model you use, because the rodent mightn't be the ideal model for every given receptor type. And finally, one of those, apart from the high resolution, the fact that you can look at very many different kinds of receptors, this method is absolutely quantifiable, okay? And that's something, for example, that immunohistochemistry doesn't enable you to do either, but here it is. And we know that the brain has very many different types of neurotransmitters and receptors, so obviously you'd want to analyze as many different kinds of receptors as possible in one sample to get like a comprehensive view. And when I first started doing this, I was looking for ways to visualize this relationship in the differences in the receptor densities of different receptors, and I came up with this polar coordinate plot. And because for the human eye, it's a bit difficult sometimes to detect patterns when you've got like clouds of points, I joined the points with these lines, and then just to make the plot look a bit tidier, I took the dots away, and that's how this plot here was born. And then Carl Sillis gave it the name receptor fingerprint, and that's how the receptor fingerprint was born. And because different areas have different receptor architectures, their receptor fingerprints will be different. So every single cortical area will have a unique receptor fingerprint. And these fingerprints are basically just numbers, so we can use them for statistical analysis, okay? And if we want to use animal models for science, you have to also know your animal model, right? So that's why in my group over the last years, we've been dedicating quite a lot of time to the analysis of the receptor architecture of the macaque monkey brain. And this is basically work that has been done by Luthia and Mei-Chi to, well, they started life as PhD students in my group, and they're now postdocs, but they're still with me for a while, and that's really, really nice. And also that they've mapped the cortex. And we also have a collaboration with Carmen Cavada at the Autónoma University in Madrid, and her PhD student Isabel is mapping the receptor distribution in the thalamus. And we are creating a 3-dimensional map which will integrate all of this information. The rotating video that you can see are projections of all the areas that we've identified on the Yerkes 19 brain. So you can use these delineations if you want to have an architectonically informed analysis of functional imaging data. And it's all in the MeBrains Atlas space. Right, so after this very fast introduction into the method and the principles, I want to share some work that Mei-Chi has been doing with you, okay? And she has been concentrating on the macaque parietal lobe, and you might wonder, well, why do the macaque parietal lobe? I mean, Brodmann already created a map in 1909, so surely we already know everything that's can be known about this. But the thing is, if you look in the literature, you'll see that there are very many different maps that cover different parts of the interparietal, of the parietal lobe, and these maps differ in the number and extent of areas that they they contain, right? And often there are discussions between anatomists because, well, in part there are, they've used different methods for their studies, so they're revealing different aspects of cortical segregation, but mainly the main problem that we see is the fact that this is these definitions are based on subjective criteria. So what we do is a multimodal analysis of the brain, multimodal in the sense that we simultaneously look at the distribution patterns of 15 different receptors, and we also look at cell body stained sections and myelin stained sections. And we quantify the differences that we see, okay? And this quantification happens in the way that we've got, so here a coronal section of macaque monkey brain, and I've zoomed into the relatively rostral part of the interparietal sulcus. We define traverses, so lines which are perpendicular to the cortex along a region of interest, and then we can then extract the gray values along each one of these traverses. And that's what you see here on the bottom of the slide. And these are then so-called receptor profiles from, position X and position Y. And you can see that they've got a different shape because they come from, two separate parts of the brain. We can parameterize these profiles, these curves, extract feature vectors from them. So again, we've got numbers, you can do statistical analysis. We can use then mathematical procedures to determine the degree of similarity or difference between two feature vectors. You've got the Euclidean distance, you've got the cosine distance. And at the Institute, when Axel Schleicher was developing this method, he found out that the Mahalanobis distance was the one which was best to show differences in laminar distribution patterns. And this method was developed to analyze the cortex. So you want to be able to see the different cortical layers, okay? And we don't just look at two profiles, we look at very many different profiles because we've got many here. We've got a sliding window procedure to calculate these Mahalanobis distances. So that's why we get a Mahalanobis function where each one of these peaks is indicative of a cortical border. And we can test these peaks for statistical validity. We also do this analysis on several consecutive sections. And when we find these borders at the same position, then we say, okay, this is a border. And this peak isn't caused maybe by a tear in the section that we're just looking at, or at a blood vessel, for example, okay? And then we can basically just transfer these significant points onto our section, and that's how we've got statistical validity of these areas that we've started off by looking for, by visual inspection, okay? And what you see here are examples of this subdivision of area 3B into an external and an internal component, different receptors, and also in the cell body stained sections. And using this method, Mei-Chi identified a total of 47 different areas within the macaque parietal lobe, okay? And then she wanted to use this data to provide more insight into the relationship between the microstructure of the parietal areas and brain function. And in particular, she concentrated on the somatosensory cortex, okay? Now, the somatosensory cortex is located in the most rostral part of the parietal cortex within the central sulcus, and we've got these strips of cortex which have areas that we know are called areas 3A, 3B, 1, and 2. So when moving from rostral to caudal, and this part of the cortex is what is called primary somatosensory cortex. Okay, and then we've got hidden in the lateral fissure, we've got this other part of the cortex, which is designated as secondary somatosensory cortex. But this is already, you know, a bit strange, because in other parts of the brain, there is one area that is considered the primary, you know, one primary visual cortex, at least in the primate brain. So there is a bit of controversy concerning whether areas 3A, B, and 1 and 2 are all can be considered primary somatosensory cortex, or maybe just one of them. Some people say that only area 3B can be considered the true primary somatosensory cortex. So Mei-Chi extracted all of the receptor densities, got the fingerprints, and then she performed a hierarchical clustering analysis, which is a statistical descriptive a descriptive statistical method which enables groupings of elements based on their degree of similarity. And the result of this analysis is like this clustering tree that you see here. And when you read it from left to right, areas that are joined together closer, it means that they are, when you're using fingerprints, they're neurochemically more related to each other, right? And what she found is that we do have a segregation of all the areas that belong to the secondary somatosensory cortex and of the primary somatosensory cortex, but actually between this or within the primary region, we have a segregation of area 3B from the rest of the so-called primary areas, okay, which points at the fact that, okay, this one area, which has been called the putative true primary somatosensory cortex, is different from the other areas. Now, she took her analysis one step further, and this is this is new stuff. The manuscript is in preparation. What Mei-Chi did was she took the receptor densities that we've extracted, not just from a parietal area, cortex, but from all of the areas of the brain that we've analyzed. And the only part of the brain that we haven't analyzed yet is the temporal lobe. So that's why we didn't include it in our analysis, because we don't have any receptor data for it. And with this information, you know, she's got the relative densities of different receptors. She then used functional imaging data, resting state functional imaging data that's freely available via the PRIME-DREE database, and tract tracing data, because we wanted to look at differences between, you know, receptor densities and structural connectivity patterns and the functional connectivity patterns in the brain, right? And with this data here, she could compute receptor covariance matrix or a structural or a functional connectivity matrix, right? And with this data here, she can do statistical tests. Now, for example, one thing that she could do is compare the relationship between receptor covariance patterns and structural connectivity patterns. That's this plot at the top, and you'd have the coupling between receptor covariance and structural connectivity, and do the same thing for the functional connectivity patterns, right? And in both cases, she found a positive correlation between the measures, but the slope was much steeper when comparing receptor covariance and functional connectivity patterns than with the receptor and the structural connectivity patterns. Now, let me show you the receptor covariance matrix just to explain a bit more of what we've got here. Mei-Chi grouped the parietal areas into different groups, which received, you know, color coding depending on their hierarchical processing level. So, for example, you know, primary somatosensory. Oh, you can't see it so well here, but this primary somatosensory here at the very top of the slide. And then we've got the secondary somatosensory here, and interparietal, superior parietal, and inferior parietal lobe areas. And then along the y-axis of the matrix, we've got all the other areas that we've mapped already. So prefrontal areas, visual areas, cingulate areas, okay? And what she found was that covariance was basically strongest between areas that were anatomically close together, but it wasn't restricted to areas that are anatomically next to each other. We also found a strong covariance with areas that are anatomically further away in the brain. And if we just concentrate here on the primary somatosensory areas, we found that the similar covariance values for areas that belong to the same hierarchical processing level in different functional systems, okay? Or we found, for example, that parietal areas were more strongly associated with visual areas and with other parietal areas or posterior parietal areas than with the primary somatosensory areas. Another way of showing receptor covariance patterns, and which was also useful in establishing this difference between area 3B as the true somatosensory cortex as opposed to areas 1 and 2 and 3A, is that they differ in their receptor covariance patterns. So, for example, we found that subdivisions of 3B have a much stronger receptor covariance pattern with primary visual areas than the rest of the primary somatosensory areas, and the other primary somatosensory areas had a stronger correlation with areas of the prefrontal cortex or of the inferior parietal lobe than do the primary, so the 3B area, okay? And then if we compare the receptor covariance matrix with the functional connectivity matrix, what we see is a similarity in both cases is that the covariance patterns are is highest with neighboring areas, but we actually find more differences than similarities. And one difference, for example, between the receptor covariance and the functional connectivity matrix is that there's a higher strength in the receptor covariance distribution patterns, but we've got more negative connections with the receptor connectivity than with the functional connectivity patterns. That's one difference. Another difference is this strong connection with the visual areas that I just described, whereas the functional connectivity, actually, there's a stronger relationship with motor areas. And then another difference between these two measures of cortical segregation is the fact that if you look within a given group of areas and you look at their connectivity or receptor covariance patterns or their functional connectivity patterns, we find a greater degree of heterogeneity in the receptor covariance patterns than in the functional connectivity patterns, okay? We can also look at receptor covariance and the structural connectivity here, because the granularity of the track tracing data that's available is slightly lower than the granularity of our receptor covariance patterns. We had to merge some of our areas together, so that's why this is a simplified receptor covariance matrix, and we can just subtract the two matrices and get this third one here, where white cells are telling us that the area in question does not differ in its receptor covariance or structural connectivity pattern, and the red or the green is telling us that we do have differences. And what we find here, again, when we compare the receptor covariance and the structural connectivity pattern, is that we've got a strong connectivity with neighboring areas, okay? In both cases, we've got the difference that our receptor covariance patterns, the strongest correlations are found between areas of the same hierarchical level, whereas in the structural connectivity matrix, the strongest correlations are found with areas of the primary somatosensory cortex and the other somatosensory areas. And interestingly, different to what I just described before concerning, heterogeneity of the patterns when you look, within an area, we found that the receptor covariance matrix was more homogeneous than the structural connectivity pattern. And we still don't understand what the meaning of that is. And then a difference is that for the a further difference, is that for the receptor covariance matrix, we have these positive correlations with visual areas, and for the structural connectivity, we have a stronger connectivity with the motor cortex. Now, I asked Mei Qi to create this figure here, which basically summarizes all of her results, plus what's known in the literature about connectivity between somatosensory areas, and where when you read it from left to right, you've got increasing hierarchical processing levels, okay? And she created this figure, which looks really nice, but she's so good at creating figures. So she then also created this other figure here, right? Which she said is just visually much more attractive than this boring figure that I asked for. Here you see the main areas that are involved in somatosensory processing. The different colors indicate different processing levels, and within each color you will see that there are different degrees of purity of the color. So when you've got a true red, for example, that is a lower hierarchical level than a color with lower purity, okay? And information first, somatosensory information first arrives at area 3B, okay, and from area 3B it is transferred to areas 1 and 2 and 3AL. These are all primary, so-called primary somatosensory areas. It is then transferred on to the areas of the secondary somatosensory cortex. Here, these green arrows in the figure, and also to higher order unimodal and also multimodal somatosensory processing areas. And then in a third flow of information, it passes on to all of these areas of the posterior parietal lobe, which are the ones then which really truly enable this integration, because these are multimodal areas, and they're the ones that truly enable the integration of somatomotor and visual information, which, for example, give us the ability to coordinate movement, to reach somewhere, to grab something or pick it up delicately, and so on, okay? So this is one thing that Mei Qi did, basically concentrating on the parietal lobe. And the other part of my talk, which is much shorter than Mei Qi's one, is a study that we performed with Sean, who at the time was working in New York at Xiaojing Wang's lab, and he was a postdoc there, and he's now moved on to create his own group in Bristol University. And what we there wanted to do was not just look at receptor densities and what their distribution in the brain is telling us about organizational principles, but also establish relationships between these receptor densities and structural parameters of the brain, such as, for example, density of neurons, or the shape of the dendritic tree of these different neurons, or also hierarchical processing levels based on analysis of track tracing data. Okay, so integrate all of this kind of information into one scaffold to enable the analysis. And we performed a principal component analysis, where what we found was that when you look at the first principal component analysis, it is separating areas that we know are involved in sensory processing. So for example, here you've got the primary visual cortex and association areas, so multimodal areas. That this segregation is basically due to the fact that we've got increasing number of receptors, okay? When you move along this here. And the second principal component is separating areas from different evolutionary origins. So we've got evolutionarily older areas. So for example, area 24 is pro-isocortex. It doesn't have the 6 layers that are typical of the neocortex. And down here, we've got the newer areas. We've got different isocortical areas, okay? And the key factor that is separating the areas along this second principal component are changes in the 5-HT1A receptor. If you look at it here in this older area, you will find that the 5-HT1A density is such that it is like a peak sticking out of the receptor fingerprint, okay? But in the newer areas, the 5-HT1 receptor density is much lower. So it appears as an indentation in the fingerprint, okay? Now, people who do functional imaging, they are interested in functions, and we want to find also the link between these receptor changes and the functions. So in cognitive neuroscience, you often hear from different networks, and I've listed a few here. The different colors of the networks and the colors of the names of the areas are chosen in such a way that the color of the areas codes for the network to which they are normally assigned, okay? And what we found here is that the principal component of our analysis didn't just segregate sensory from association areas, but it also segregated areas that are involved in sensory processing from areas that we know that are involved in higher order cognitive functions, okay? And when we look at the second principal component, we've got down here in this bottom part, we've got areas which are involved in the so-called dorsal attention network, whereas up here we've got areas which are part of the default mode network, so the one which in theory is monitoring what is happening inside us. So here we've got a segregation between areas that are more involved in internal focus versus external focus, okay? And then the other thing that we wanted to see was also, well, how do these differences relate to these different structural aspects? So what we found was that there's a positive correlation between these, for example, the first principal component and receptor densities. So we found more higher receptor densities, the higher our first principal component values become. We also found a positive correlation between receptor densities and synaptic density, but we actually found a negative correlation between receptor densities and myelin density, okay? Because which is sort of like intuitive, because if there's myelin, you don't have synapses, because that's the part of the neuron where you want the electrical impulse to be transferred along without any kind of you want a fast transmission without any kind of modulation via you know, neurotransmitters. And to finish, I want to show you an image which I found fascinating. Sean projected the first principal component onto the surface of the Yerkes brain, okay? And when I first saw this image here, it reminded me of this image here. And the image on the right shows you the human functional zones, which Marcel Mesulam defined, you know, based on like a lifetime of research that he'd done, plus more that he's done after that. But I mean, it was the fact that if you look, for example, at this blue zone in the human brain, where you've got the function of somatosensory motor cortex, it correlates beautifully with this blue part here of the principal component values in the macaque monkey brain. And you know, I could go on for a long time looking at these different things, but in the interest of time, I'd just leave it at here and say that for me, this was a validation of the fact that receptor fingerprints really do help us study the relationship between structural and functional segregation of the brain, and also that the macaque monkey brain is a really good model for neuroscience to understand how the human brain works. So, and with that, I would like to thank you all for your attention. And of course, you know, all my team members, my collaborators, all my, you know, co-authors and so on for all of their support.
I guess we have time for like maybe 2 questions. We'll see. It depends. Does anybody want to start?
Just touching on like actually the very last slide there on the 3 associated divisions. So I'm saying the biggest difference between the monkey and the human would be like we have more space allotted to these associative cognitive areas compared with the monkey. So based on your like findings or research, what do you think are the driving forces behind that like reallocation of cortical area?
Well, the thing is, these prefrontal areas are also the ones that have undergone the most changes in evolution and the greatest degree of expansion. Prefrontal cortex and also here the parietal lobe. Those are the 2 parts that have changed most. So it's not surprising that they're relatively small here. I mean, basically, it's a combination of factors, I'd say. It's the fact that when you've got 2 areas that can be considered homologue between both species, relatively speaking, they would be larger in the human brain than in the macaque monkey brain. But I think also we've brain areas that didn't exist in the macaque monkey brain have emerged through evolution. Just the same as when you look at very primitive, like insectivores, for example, they actually don't have separate motor and somatosensory cortex. They have this somatomotor amalgam. Well, there are areas in the monkey brain which have separated into or resulted in different areas in the human brain.
So much excellent work and great job. I'm wondering about like treatment with like for example disorders like ADHD. Have there been studies which show how say a lifetime of treatment affects say the activity and the organization and the function versus no treatment?
They definitely will. The receptor densities that you have, it's not a static situation, so they do change through time because of function, but also due to medication or whatever party drugs. So that can change the receptor, or not can, it does change receptor densities. We know that very many psychiatric diseases and neurological diseases are actually related to imbalances in receptor densities. And medical treatment aims to, well, counteract these imbalances, sometimes with more success than others. I can tell you, for example, that even short term, so I was involved in a study of brains that were obtained from patients with hepatic encephalopathy, and in the final stages of the disease, they were in coma, and some of them had to be intubated, so they had to undergo sedation. And when I looked at the densities of the GABA-A associated benzodiazepine binding site, which is the one which is targeted by the sedative, it was significantly higher in the brains of the patients that had been sedated because the patients that needed sedation because of intubation, as opposed to the patients that were in coma but didn't have respiratory problems, so they didn't need this artificial ventilation. So it definitely affects it. And in ADHD, for example, it will, I don't know specifically of any studies. I think there's some mouse models for ADHD. I know of mouse models for autism, and the receptor architecture of the mouse brain, the knockout mouse, is different from that of the control mouse. You promised only easy questions, Yasha. Remember.
The real question, more. I'm curious.
Yes.
Because you started with something that is very interesting, why we just don't connect neurons, because they have different speeds. We can control the speed across these receptors. And when we go to the gradient, or we go to the very nice figure that you asked, also the figure that Mechi made, is the speed the factor that causes the hierarchy, or is it can be any receptor, the fast and the slow go together?
The fast and the slow can go together.
So the speed is not what is making controls, and we don't have like, we are controlling associative areas faster or the sensory faster. As we go to associative, we get the slower ones added.
That's a very good question. And the answer is I don't know, because I didn't look at it. But this is being recorded. Yeah. So I'll tell Mechi to look at the video. Yes, it's a very interesting question, and I don't know. We want to look, when we have the 3D reconstructed receptor autoradiographs, analyzing all of this in these 2D sections is super time consuming. And yeah, sometimes you don't have enough data. What I want to do is look at ratios between not just excitatory inhibitory receptors in the sense of GABA and glutamate, which is what people very often think about when you talk about excitation inhibition. They think GABA glutamate, end of the story. But divide the modulatory neurotransmitters into the excitatory and the inhibitory neurotransmitter receptors, because they're the ones that I think are responsible for the fine-tuning of brain activity. And then it would be interesting to see if we find differences between primary areas or unimodal areas and multimodal association areas. Yeah.
It wasn't.
Thank
you so much.
It wasn't an easy question, but it was really nice question.
Thank you.
A pleasure. Thank you for the invitation.